Hello and welcome to the immunology series of Gain Family and myself Arijit, and this lecture series will be primarily focusing on MSc entrance exams and a lot of MSc entrance exams like JAM or BHU biotech entrance exams and others. They focus a lot of questions on immunology, so it's better to understand the immunology very well. And immunology is a complex intricate subject and there are a lot of things and all of them are interconnected, so it's better to understand the logic of immunology and logic behind those things first, and then to mug up all the intricate details. Okay, so this is actually, I'm calling it layer 1, and this lecture will be laying the ground for further intricate details, and this will be the very fundamental things about immunology. And so, as we know that we all have read or seen the comics of Batman, but there is no importance of Batman without Joker, right? So there is there is no importance of immune system without antigen. Okay, so we need antigen for the immune system to be of any importance. So what are antigens? So antigens are mostly protein molecules, okay, but there are certain antigens which are lipids or glycolipids or carbohydrates, but let's focus on this protein part now because most of the antigens are proteins. So these antigens, when they get into human body, okay, so they face a judicial system. Okay, so they face a judicial system, and this system is a part of immune system of ours, and this judicial system actually detects all the protein in our body that is roaming around in our body, and they give a verdict whether that protein is good or bad. In immunology terms, that is whether that is self or whether that is our own protein or that is foreign. Okay, so if this judicial system gives a verdict that one certain protein is a foreign molecule and came from outside, and that kind of terrorist molecule will be considered as bad for our system. Okay, so that needs to be taken care of. So what is this judicial system of ours? So this system is actually the APCs or the antigen presenting cells. Okay, so as the name suggests, they actually present antigen on their surface so that the other effector cells which can take care of the antigen, they can recognize that antigen. So I'll be coming to that. So APCs are mostly of three types. The most efficient one is the dendritic cells. Okay, dendritic cells, and a bit less efficient is macrophages. Okay, in short we call it as M5 and macrophages, and the least efficient is B cells. But we need to remember that the primary function of B cell is to produce antibodies. Okay, sorry. The primary function of B cells is to produce antibodies. So now what this APC, let's say dendritic cells, does. Okay, so let's say this is the protein molecule or the antigen that came inside our body. So what this dendritic cell will do is it will take up the antigen. Okay, through the process of phagocytosis. Through the process of phagocytosis, it will take up the antigen, and the antigen will get into a vesicle. The better term is phagolysosome, because with the phagosome that is containing this antigen, the lysosome gets fused with that vesicle and it produces phagolysosome. We'll cover all these details later on, but let's say for now this antigen is inside this vesicle, and inside this vesicle through lysosomal enzymes this antigen molecule gets chopped off in smaller pieces. Now what is the reason for this? The reason is this antigen needs to be displayed on the surface, right? But it to display this, this antigen has to be put on a cup or say a pot. Okay, right? This antigen has to be put on a pot and then has to be displayed. But APCs, our APCs, none of them has this kind of big pot to actually hold this big antigen, okay, or this big protein molecule. So it actually chops off this big molecule into smaller pieces so that whatever smaller sized pots are there inside the APCs, that can actually hold this smaller pieces of antigen, okay, or the fragments of antigen. And now this whole thing with the pot and the smaller fragment, that gets transported to the surface and then it gets displayed on the surface. Now these pots are where the antigen is actually placed, that is called MHC, or the major histocompatibility complex. So we'll see this in later videos. And when this antigen fragment is displayed on the surface of APCs, that is actually recognized by a particular cell that is called T cell. Okay, this T this T stands for thymus. Okay, and why it is called T cell we'll see later. But when this T cell recognizes this antigen along with some other cells, they take care of this antigen. Okay, and this is the central dogma or the fundamental basis of immunology. Okay, if you know this, then in later videos we will see all these details. What are the properties of antigen, when a protein molecule can be called an antigen, okay, and when how this antigen gets internalized into APCs and how this fragmentation occurs, and how many types of MHCs are there, how many types of T cells are there, and what are these other cells, and how they take care of this antigen. Okay, and this antigen can be protein molecule or carbohydrate or lipid, but mostly they are protein molecules. Okay, so this is for this lecture or this video, and we will see the later details in later videos. So do like our videos and do comment how did you feel about it, or whether you liked it or not, or any suggestions. Okay, and do like our page obviously. And so for now, bye, and we'll see in the next video.